വൈറസ് കൂടി കോവിഡ് വൈറസ് കൂടി ആക്രമിച്ചാലുള്ള സ്ഥിതി എന്താകും? അതിനെ പറ്റിയിട്ടായിരിക്കും നമ്മൾ ഇന്ന് ഏറ്റവും കൂടുതൽ നന്നായിട്ട് ചർച്ച ചെയ്യുന്നത്. നമുക്കറിയാം, ക്യാൻസർ എന്ന് പറയുന്നത് ഒരു കഠിനമായ രോഗമാണ്. ആ രോഗത്തോടൊപ്പം ഈ കോവിഡിന്റെ ആക്രമണം കൂടിയാകുമ്പോൾ വല്ലാതെ രോഗികൾ തളർന്നു പോകാം. അങ്ങനെ തളർന്നു പോകുന്ന മനുഷ്യരെ നമ്മൾ കൈപിടിച്ചുയർത്തുന്നതിന് ഈ വെബിനാർ പ്രഭാഷണം അല്പമെങ്കിലും സഫലമാകുമെങ്കിൽ നമുക്ക് അതിൽ കൃതാർത്ഥരാകാം എന്ന് മാത്രം പറഞ്ഞുകൊണ്ട് ശ്രീകുമാർ പി ചെല്ലപ്പൻ സാറിനെ ഏറെ ആദരവോടെ പാരമ്പര്യവും പ്രൗഢിയുമുള്ള കൊല്ലം ശ്രീനാരായണ കോളേജിന് വേണ്ടി ഞാൻ ഒരിക്കൽ കൂടി സ്വാഗതം ചെയ്യുന്നു. നന്ദി, നമസ്കാരം.
Thank you very much, Sunil Kumar sir. With great pleasure, I am introducing our most respected resource person today, Dr. Srikumar P. Chellappan. Currently, Dr. Srikumar P. Chellappan is a chair of the Department of Tumor Biology at the H. Lee Moffitt Cancer Center and Research Institute at Tampa, Florida, USA. Dr. Chellappan obtained his BSc and MSc degrees from the University College, Thiruvananthapuram, and his Ph.D. in biochemistry from the Indian Institute of Science, Bangalore. Subsequently, he did his postdoctoral training at Duke University Medical Center, Durham, North Carolina, and became an assistant professor in the Department of Pathology at Columbia University in New York in 1992. He joined Moffitt Cancer Center in 2001, where he is a tenured professor and senior member. Moffitt is the third largest cancer center in the US by patient volume. He was recognized as the Scientist of the Year at Moffitt in 2007 and as a Moffitt Distinguished Scholar in 2015. He was also presented the Outstanding Achievement Award by the Society of American Asian Scientists in Cancer Research in 2016. Dr. Chellappan's research interests include signal transduction and transcriptional regulation, especially in the context of smoking, cancer stem cells, cancer drug discovery, tumor progression, and metastasis. His research has been continuously funded since 1994, and his work has been published in journals like Nature Communications, PNAS, Journal of Clinical Investigation, JNCI, Stem Cells, Cancer Research, etc. He is currently an associate editor of Molecular Cancer and serves on the editorial board of Neoplasia and PLOS One. In the past, he had served as an associate editor for Molecular Pharmacology. Dr. Srikumar Chellappan served on more than 100 NIH study sections and grant review panels of national and international agencies. A very warm welcome to you, sir, to our webinar series. This is the 25th talk. Over to Srikumar, sir.
Thank you. Thank you very much for the very kind introduction and for the invitation to speak in this webinar series. So I thank all of you for attending in such a rainy evening, and I should give a special thanks to the principal, Dr. Sunil Kumar, for being here when there is no power at his home and giving a very nice introduction. I am not too sure whether my talk will live up to his expectations, because the COVID aspect is not that prominent in my talk, because the problems in cancer treatment and therapy, they exist whether there is COVID or not. Thank you so very much for the introduction, for the kind words, and for this opportunity to talk in this venue. I attended some 2 or 3 talks in this series, and they were all amazing, very captivating, and I really don't know whether my talk will measure up to those levels. The only problem has been that it is 9:45 in the morning here. We are all starting our work days, and your times exactly correlate with our busiest time of the day. So I couldn't attend many of the talks in your fantastic seminar series. So it's a great opportunity to be here, and me being from Kerala, it is very disheartening to see all the problems and issues there with COVID, as well as and currently with the weather. So let me start sharing my screen and go over my presentation. I should be probably done in 45 to 50 minutes. I'll be talking the first half almost about the general principles of what is happening in cancer, how is COVID is affecting it, etc., and what are the current problems, and some of our own work in my lab that is going on, and how we are trying to address these issues, and how we are trying to develop novel therapies for cancer. So with your permission, I may share my screen now. I hope you can all see this.
Perfect, sir.
Okay, let me see if I can have. Got it. So this was the title of my talk, and so essentially one of the basic questions, which I know the audience is very varied. There are a lot of students I have seen from the field of arts, commerce, and non-science fields. So just to make things very easy for them also, I'll try to give it a very basic introduction. So essentially one of the basic questions has been, how does cancer develop? The bottom line is that cancer is a genetic disease. It is caused by changes to genes that control the way our cells function, especially how they grow and divide. Genetic changes that cause cancer can happen because of errors that occur as our cells divide. As all of you know, DNA is a major genetic component that governs our heredity as well as other functions. So errors that can happen when the cells divide in the DNA replication that can cause cancer. Damage to DNA caused by harmful substances in the environment, such as chemicals in tobacco smoke, ultraviolet rays from the sun, and many other mutations that were inherited from our parents. All these things can contribute to the development of cancer. As you can see, there is a chromosome, the DNA here. There are proteins associated with the chromosomes. So in addition to these genetic aspects, there could be epigenetic aspects as well. So which are the inherited changes in the gene expression patterns, but that is not a genetic change, that's not caused by a mutation. So these happen by modifying these proteins that are associated with the DNA, or by modifying the DNA itself without mutating the sequence. So those are some of the epigenetic changes. So there are two major classes of genes that essentially regulate the formation of cancer. One group is the oncogenes, other group is the tumor suppressor genes. I think many of the science students may know all this, but I'm just saying for everyone's sake. So oncogenes are the proto-oncogenes, etc., which when mutated, mutations in this class of genes essentially causes the proliferation of cells, causes a tumor to migrate, go to other locations to metastasize, etc. So oncogenes are almost like the accelerator in your car. I mean, so if you put a stone on the accelerator, that's what happens. It is equivalent of that. When oncogenes are mutated, the cancers grow very fast. Tumor suppressor genes are, as the name suggests, they actually prevent the cell proliferation, they prevent the migration of cells, etc. So they're almost like the brake of your car. It's almost like instead of the accelerator getting stuck, a mutation in the tumor suppressor gene is almost like a brake being lost. So there's this very fine balance in a normal cell between the oncogenes and the tumor suppressor genes that regulates the normal proliferation and growth of cells. So when this balance is affected, either by mutation in oncogenes or in tumor suppressor genes, the balance of the proliferation is lost and the cells develop cancer. So there are different stages of cancer, which you may see in a different diagram, different pictures. So this is almost like a epithelial cancer, ductal adenocarcinoma. So these are cancers, the type of, one of the most common type of cancers. Breast cancer come under this category, pancreatic cancers, lung cancers. So there are these ducts which are covered by epithelial cells, epithelial cells that form a layer on top. So cells with mutations, they start as the initial genetic changes, a mutation in an epithelial cell. Then it develops into hyperplasia as the time goes by, accumulation of more mutations or contribution of the cell surrounding cells. Hyperplasia is a situation where there's uncontrolled cell division. This is still not cancer yet. This is almost like the warts or Sherikolla cancer. From there, it goes to dysplasia, which is in a very early stage of cancer, we can say. The cells not only divide, but they change shape, they start moving around a little bit there. Their whole biology is starting to change, and they form cancers in situ. cancer in situ. That's where the cancer in the same location. So the cancer cells stay in place. From this, there is metastatic cancer where the cancer cells can migrate. They become highly invasive. They have angiogenesis in them. You get supply of blood vessels. The tumors get into the blood vessels, go to distant locations, and there is metastatic spread of malignant cancer. So this is the latest stage, the most advanced stage of cancer. So once it reaches this stage, it is challenging to overcome that, but nowadays there are ways to combat that. But if the cancer is detected in one of the earlier stages, it can be, the chances of getting a complete cure is very, very high. So this is a very, very basics of that. So what happens during this process? Just to show you a little bit of science. So this is a normal epithelium, the duct, whether it is a mammary duct or the pancreatic pancreas duct, etc. There's a basement membrane. The epithelial cells are sitting around, acquire mutations, they become dysplastic, they change the shape. Then they undergo another process called epithelial mesenchymal transition. These flat, well-organized cells, now they become spindle-shaped. They can move around and they enter the blood vessels and they go to the secondary site as cancer metastasis or the spreading of cancer. So all these changes are governed by expression of genes and mutations of genes. So these cells have an expression of a classic set of epithelial genes that allow the cells to stick to the epithelial, stick to the basement membrane. But changes in the gene expression patterns allow these cells to change their shape and they allow them to migrate. By this time, all these genes are shut down, they don't express anymore. You get a totally different set of genes that change the shape of the cells and allow its mobility from one place to the other. These genes allow them to enter the blood vessel, survive in the blood vessel, get out of the blood vessel, and essentially grow as a secondary tumor in a metastatic site. So after this, so what is happening in the cancer incidence in the United States and India? So, I mean, we always hear that the cancer incidence is so high. We all know somebody or other, our neighbors, friends, who have cancer. But the fact is that there is a lot of cancer in the world. There's around 18 lakh projected new cases in 2021 in the US. Around 6 lakhs of people are expected to die from cancer in the US, and the numbers in India are probably much higher. But surprisingly, this is one of the promising part. There is a 31% reduction in cancer-related deaths from 1991 to 2018. In this particular period, there was a remarkable reduction in cancer-related deaths. And some similar trends are happening in India as well. There is a shift from the increase in the cancer incidence to a reduction in the cancer incidence. So it is not that dramatic as in the US, but it is going to happen in India as well. So what is the risk of developing cancer in lifetime? It is 42% in males and 37% in females. And risk of dying from cancer, 22% in males and 19% in females as of 2018. So this was the last year when the cancer statistics were available. So people have a fairly high chance of getting cancer sometime during their lifetime. So, I mean, cancer is generally a disease of the older age. So generally people in the later stage of life, they are the ones who get cancer. And essentially the old age essentially correlate with the incidence of many diseases, not just cancer. So we wouldn't worry about it too much. So in India, for example, there is around 13 lakh patients living with cancer in India in 2020. The incidence appear to increase a little bit. Market variation in incidence rates across India with maximum incidence rates in the Northeast. So in India, as far as I know, there was not much infrastructure to monitor the cancer patients, collect the samples and study the cancer, for example, till 15 or 20 years ago. Now there is a very strong, very effective network established. And I think there's a branch even in Kollam where they're collecting samples, they're monitoring the incidence of cancer, what is the progression, what is the survival, et cetera, et cetera. So, and what they're finding is that there are some very nice review articles and there's an annual report coming out, which shows that the variation is very, sorry about that. I mean, the variation in the cancer incidence is very remarkable. In some parts of India, there is very little cancer incidence, whereas certain parts of the northeastern India, the cancer incidence is very, very high. Major sets of cancer in India are the breast, lung. These are the same in the US as well. Mouth, cervix, uteri, and tongue. The mouth and the tongue, they are generally correlated with smoking as well as chewing of tobacco and murka and lung. That's why the mouth and the tongue cancers, oral cancers are appearing in India. But generally, there is a very promising trend in the occurrence of cancer nowadays. So what is causing this particular reduction in the incidence and mortality? The major change that is happening is that the cancer education and awareness. I mean, compared to even 20 years ago, we are all much aware about what we eat and we don't smoke and things like that. So the cancer education and awareness has made a very significant impact on the reduction in cancer-related mortality. And again, such education and awareness has caused us to improve our lifestyles. We try to walk more, we try to exercise more, which causes a reduction in not only cancer, for cardiovascular diseases and so many other diseases. So the lifestyle changes, which is a result of this cancer education and awareness, has played a very major part. Another very major component is screening and early detection. Now, mammography and screening for early detection of cancer, that is very common. In India, I mean, that's something done fairly routinely among the people who are interested, and there are access to these things, access to better cancer care, access to the screening and early detection. Early detection especially plays a very major role in the improvement of cancer therapy and the cancer response. And access to better cancer care. I mean, when I was in India, I left India 34 years ago. There were not too many cancer hospitals, not too many private hospitals compared to 30 years ago. Now there are hospitals everywhere, and you get first-class cancer therapy in India as you get fairly comparable to what you get in the US. I think many of the doctors in the group who may be attending may know that, but in India also, they follow the same criteria, the same treatment plan as they follow in the US. There is a website and there's an organization called National Comprehensive Cancer Network, NCCN. NCCN has these guidelines for what are the therapies to use for what type of cancer, stage 1, stage 2, stage 3. And what is being followed here is pretty much the same what they follow. They follow the same NCCN guidelines back in India as well. And another major impact, of course, is the development of novel therapeutic strategies. I will come to that. I will talk about that fairly soon. So these have caused a major reduction in the incidence of cancer in the US, at least a major reduction in the incidence, even though the actual numbers seem to be pretty high. And I'm sure it is going to happen in India as well as time progress. Usually India lags behind by 4 or 5 years, so it is going to happen there as well. So these are all very, very promising aspects of cancer therapy, as we can see. So just to have 2-3 slides, I have just, I think, 3 slides on how COVID has impacted all this. So COVID, as you know, has been prevalent for a year and almost a year and a half. I think it was first reported in India back in February of last year. In the US also, it started appearing in the February of last year. So it is around 16 months. So there have been a few relevant papers on population-based studies on the incidence of cancer in cancer patients. I'll come to that in the next slide. So this was an article in the put out by American Cancer Society, which is one of the major organizations in the world that follows cancer treatments and the epidemiology of cancer, how widespread is cancer in the society and how they respond. So what they are finding is that what is the potential impact of COVID-19? So they just published this 2-3 months ago, I'm sorry. One of the major issues that is affecting the cancer outcomes is a reduced access to cancer care. So fear of infection, allocation of health resources, unemployment leading to financial insecurity and insurance loss, shutdown and social distancing. So even in our cancer center here, when the COVID was at its peak last March till July, the cancer center hardly took any new patients. There were hardly any cars in the parking lot. So the patients were they were doing telemedicine and there was not much access to the actual care what they received. So this is going to cause a problem as the time goes by, but I think that is over now. the prevention and early detection, that has been affected by COVID. I mean, all around the world. So the preventive visits have been reduced, screening, especially mammography and pap smears, all those have been reduced significantly. Abnormal test follow-up, symptom follow-up, all these things have been delayed due to COVID, at least the past 5-6 months, but things are starting to pick up again, at least here. So diagnosis, later stage diagnosis, because of the lack of preventive visits or the delay in the preventive visits, the cancer diagnosis is happening at later stages. So year ago or 2 years ago, a cancer that was diagnosed in the stage 2 or the stage 1, now because of the few months delay in the screening and the diagnosis, the cancers have probably progressed to a later stage. So later stage cancers, as I mentioned, they have lower probability of survival, fewer treatment options, more intensive treatment. All these things are needed for that. And the treatment itself, delayed and modified treatment, so that is happening fairly often. Postponed surgery, radiation and chemotherapy, less intense chemotherapy, and non-standard care. All these things are happening. So they are expecting an increase in cancer-related mortality because of these particular COVID events, which will come out in the next year's survey. So this is something that is fairly bad that is happening, or that is a potential impact of COVID on the actual cancer outcomes. Especially, I know that I mentioned the next slide, but one of the major things that is happening is that some people are putting off chemotherapy. They are taking a break in chemotherapy, for example. I'll come to that. So what is happening, or what is the incidence of COVID in cancer patients? There are two totally different points of view. There's a whole bunch of earlier studies that was conducted in the middle of year 2020, in the last May, June, where they showed that there was an increased incidence of COVID-19 in cancer patients, especially those with hematological malignancies and lung cancer. So these are leukemia, lymphomas, etc., MDS. Those are the blood cancers. That's what it is. So those are lung cancer patients appear to have an increased incidence of COVID-19. They also suggested that cancer patients are more susceptible to develop more severe symptoms. That is understandable because patients who undergo chemotherapy and are immunocompromised. So chemotherapy, I will come to that in a minute actually. Chemotherapy essentially kills a lot of normal cells in the body. And people undergoing chemotherapy have less immune response. So that is not the best situation to combat COVID. So patients who undergo chemotherapy generally in the earlier studies showed that they are prone to more severe symptoms of COVID, or they get a more serious version of COVID if they're undergoing this. So this was another important point. Delays in obtaining therapy, breaks in ongoing therapy, etc., enhance mortality. That is something that you can imagine. And people had shown that a break of 4 weeks of treatment of cancer, of radiation, chemotherapy, etc., that might increase mortality up to 20%. So COVID has really affected at least a subset of patients whose therapy, whose routine therapy has been interrupted by the COVID. So those are all some of the major findings that were made in almost a year ago, or at least 6 months ago. So what is happening now? So more recent findings. So they did larger studies on larger patient populations. More recently, I mean, publications in 2001 and 2021. I'm still talking of 2001, which is more what I've used. 2021, February, March, and the very recent paper, study from the ACS. I given some links at the bottom here. What they're finding is that they did not find a huge increase in the incidence of COVID in cancer patients compared to normal population. So there was one study that said there is a 4%, the cancer patients have a 4% higher chance of getting COVID, or there are more 4% more cancer patients who got COVID compared to an equal number of normal patients, but that has been debated. So it is not a significant increase. Not only that, the results of the earlier studies and the current studies are confounded by the fact that both the diseases are more prevalent in patients with the same underlying comorbidities. I mean, as our respected principal mentioned earlier, there are a lot of underlying diseases. I mean, diabetes, old age, immunodeficiency, obesity. These are all lifestyle diseases or other problems which people already have, the confounding factors of the comorbidities. These are pretty much the same for cancer as well as COVID. So now the problem has been to distinguish, is it something specific to cancer or is it something specific to one of these comorbidities? So that is where the results are not fully conclusive yet. But it seems that there is no remarkable increase in the incidence of cancer among COVID patients, which I think is highly promising, and it is I mean, a beacon of light in this particular darkness of COVID. I think that is something pretty good. So overall, it was found that cancer patients did suffer from severe symptoms of COVID-19, and this is perhaps related to immunodeficiency. Many of the cancer therapies called the normal immune cells, and if you're immunodeficient, of course, you have more severe symptoms of COVID. So these are some of the citations I had put, which I can send to anybody who is interested. There are so many more papers in this field that has come out in the last couple of months. They all point to the same thing. I mean, cancer patients, they don't have to worry too much about catching COVID. Just follow the normal precautions, mask up, be careful with whom you are talking to, socially distance, take your medicines, and just be careful. I mean, that's all they are saying. So that is a good thing about it. So having told all this, let us talk about cancer therapies a little bit. So what are the common cancer treatment strategies? I believe most of you know about all this. If a cancer is localized, the earlier stage of cancer, surgery is the most advisable and the most widely used strategy. So if a cancer can be surgically removed, one should definitely go for it. That is probably the most important and the most significant therapeutic advantage. Surgery is the one. And if the localized cancer, certain cancers can be treated very effectively by radiation therapy. They all have side effects and some issues. For example, for surgery, there are still cancer cells left in the margins and other regions. And some studies have shown that certain cancers, when they are surgically exposed to oxygen and they're removed, some of the remaining cells can spread to other organs. So about 99% of the time, or at least 90% of the time, this is probably one of the best therapeutic options out there. Radiation therapy similarly has side effects. It can kill normal cells. It can make people sick. It can make them nauseated, digestive problems. So those things can happen, but it can also very effectively cure localized cancers. The problem is when the cancers are already spread. So targeted therapies, I will come to that in very detail a little bit later. That's what our lab and all have been working on to create. So targeted therapies are using specific agents to kill certain cancers harboring certain specific mutations or gene alterations. So what happens, as I mentioned, a lot of the people, when they are diagnosed with cancer, it has already been spread, it has been fairly advanced. For those kind of patients, the most widely used therapy has been the chemotherapy. I will come to that. We'll show one more slide on this. Chemotherapy is the most widely used therapy to combat metastatic cancers, cancers that are spread to other places. Certain cancers can be treated with hormone therapy because certain subsets of breast cancers, prostate cancer, etc., can be treated by hormone therapy, and that is fairly efficacious. Immunotherapy, this is something that is very promising, and it is almost like a miracle. It is working extremely well in a subset of patients. I will come to that in a second as well. So coming to chemotherapy, I think this is the only slide I'm going to show on chemotherapy. So as you know, the cells divide, they go through the resting cells, they undergo cell division, they go through a growth phase, G1 phase, where the DNA replication, all the necessary material for DNA replication are being synthesized. The DNA actually replicates in the S phase, that's where the DNA synthesis takes place. Then there's more cell growth, then there is mitosis, where the actual cell division takes place. So the chemotherapy, essentially 99% of them take advantage of the fact that cancer cells divide much more profusely or at a much higher rate than normal cells. So they use drugs like taxanes, which affect cell division, microtubules, a lot of hormonal agents, Antimetabolites, methotrexate 5-fluorouracil, which prevents DNA replication. This also affects the DNA replication, etoposide. There are also non-specific agents like alkylating agents, platinum agents. All these guys can kill the replicating cells. The cells are dividing very fast. The chemotherapy agents can work quite well against them. So what is the problem with the cancer, with the chemotherapy agents? It has very strong side effects because it is not just the cancer cells that are dividing very fast in the body. There are cells of your gut epithelium, of your alimentary canal, which is dividing very fast. That's why people get nausea, they have diarrhea after chemotherapy. Your immune cells are dividing fairly fast. You are eliminating them. So that is not very good. You lose your hair because the hair follicles are dividing cells. So chemotherapy has a lot of side effects. It is highly immunosuppressive. And that is why people with chemotherapy can get other secondary diseases as well. But as of now, this is probably, or classically for the past 30 years or 40 years, this has been the most effective treatment for metastatic cancers or cancers that are spread already from one side to the other. So this is probably one of the most recent and most promising mode of therapy. So this is utilizing our human immune system to combat the cancer within. So essentially, when a cancer cell or an antigen presenting cell appears in the bloodstream or appears in the body, the T lymphocytes, which as you know are a cell of the immune system, they recognize the antigen on the cancer cell itself or on the antigen presenting cells. The T cells will find that, okay, there's a foreigner here, there is a problem here, we should take care of this. So they recognize this outsider, the non-self cell. These activated cells will expand. It's almost like calling mall security guards. They secrete a lot of cytokines and proteins like granzymes and perforin, and they kill the tumor cell. So all this depends on the interaction of the T cell receptor with the antigens present on the tumor cell. So this is what is generally happening in the normal immune system. So the question is, if you have a very strong immune system, why doesn't the tumors, the T cells, eliminate all these cancer cells normally? It does happen. Many people, we don't get cancers to a great extent because of the T cell mediated elimination of tumor cells. It is happening in our body. We all get abnormal cells once in a while, but our immune system is eliminating them. So that's all happening to a certain extent. But how do the tumors escape and form the actual tumors? So there are multiple mechanisms. So one of the most common mechanism is the fact that the T cells have certain receptors. So these are receptors that recognize the antigens. The T cells also have receptors which ask them to slow down. These are inhibitory receptors on the T cells. So what the function of this inhibitors of T cell is that once the immune response is done in a normal human being or against a bacteria or any of the non-self proteins, the immune system has to shut down. The immune activity has to be reduced so that the normal cells will not be affected. So this is essentially telling, these are the receptors that tell the T cells that it is all safe now, you quiet down, don't attack anymore. So there are all these inhibitory receptors present on T cells, PD1, PD2, CTLA4. These are some of the major inhibitory receptors on T cells. So why do tumors grow in people or in mammals? The cancer cells, they are very adaptive. They evolve very well. They are extremely smart. I will come to that a little bit later. They express ligands, or they present, instead of the antigens here, they have this proteins called PD1, which I'm showing an example here, which can bind to these receptors and shut down T cell activity. So the tumor cell by inherent mechanisms, PD1, PD2, they have multiple immunosuppressive proteins expressed on their surface, which essentially tells these T cells, don't attack me, or you cannot attack me. I have the shield around me, so you cannot attack me. So this causes T cell suppression because these receptors, inhibitory receptors, are activated. Now the tumors can grow as they want. So the question is that if you can block this interaction for somehow, the T cells can still active and you can kill the tumor, you can still destroy the tumors. So how can you do that? One of the major advance recently has been the development of antibodies against PD-1 and PD-L1. So many of the companies, especially that's what Jim Allison got Nobel Prize last year in medicine, they have found that if you can use antibodies against PD-1 on the T cells or against PD-L1, you can prevent this inactivation of the T cells. If you use these antibodies, these inhibitory receptors are shut down. Now the T cells can function like earlier and they can eliminate the cancer cells. So this is essentially one of the schematics of that. This is a tumor cell or antigen presenting cells. You have CTLA4, you have, this is the activating receptor. This is also activating receptor. These are the inhibitory receptors. So this T cell is not active. This is inhibited. This can also have, cardiotoxicity. Now you use these antibodies against PD-1, antibodies against CTLA4, which is another inhibitory receptor. All these guys, the ipilimumab, nivolumab, pembrolizumab, all these guys, or against the cancer cell PD-L1 ligand. These antibodies can keep the T cells functional. Now they can go and attack the cancer cells and eliminate them. So what are the promises of this particular therapy? It is very highly efficacious in tumors with high mutational burden, example, lung cancer and melanoma. Now, essentially, what the doctors do is that most of them, especially if people are smokers, so immunotherapy surprisingly works much better in lung cancers in smokers because they have higher incidence of mutations. So lung cancer in smokers have mutations in the KRAS oncogene, whereas the non-smokers have EGFR, mutation in the EGFR oncogene. So for lung cancer, if they find that the tumors are expressing high levels of PD-L1, you can do a quick immunohistochemistry or immunohistochemical staining for PD-L1 on the tumors. And with the patients, the tumors are expressing high levels of PD-L1, in those patients, the immunotherapy should work. So only about 35% of patients, cancer patients, respond to immunotherapy. So that is a problem. The rest is 60 to 65% of the patients don't respond to immunotherapy for unknown reasons. There is also cardiotoxicity and neurotoxicity. So those can be pretty severe toxicities that are related to immunotherapy. But this is one of the major reasons why the mortality of cancer has been reducing, especially in very, very recent times. And one of the interesting trends is that the mortality from lung cancer is not the high, or lung cancer is not a cancer that is causing the highest incidence or mortality. It is breast cancer nowadays, because breast cancer so far do not respond to immunotherapy. It is supposed to be an immune cold tumor because they don't respond to immunotherapy. So now there are so many attempts going on to enhance the response of breast cancer patients to immunotherapy. Those are studies that are going on. I hope I am making sense and it is not too confusing. So what are targeted therapies? As I mentioned in the very first slide, there are a lot of mutations that can happen due to various reasons. These mutations can alter the proliferation of cells. So what do these genetic mutations actually cause? Many of the oncogenes are essentially growth factor receptors, or those are the proteins that essentially present on the surface of the cells or inside the cells, which mutations cause them to be more active. So many of the oncogenes are signaling molecules that essentially tell the cell to divide more fast. They are the signaling molecules. There are so many tumor suppressor genes which are very difficult to target by drugs. So there have been targeted drugs against these specific mutations. For example, I mentioned EGF, epidermal growth factor, binds to epidermal growth factor receptor. So a large number of lung cancer patients, breast cancer patients, etc., have mutations in the EGFR oncogene. Same thing for HER2, which is another EGFR family member, c-KIT oncogene, c-MET oncogene. Many of these genes that are mutated can be targeted by the small molecule inhibitors. So these are also chemicals, but they are different from chemotherapy because they act only on cells, at least technically they act only on cells where these oncogenic signaling pathways are activated. So there is a Wnt signaling pathway, there is a ERK signaling pathway, there is RAF mutation. There are mutations in multiple genes down here against which effective drugs have been developed. So this would be the best case scenario. You are not killing all the dividing cells randomly. You are just preventing the division of cells that carry these specific mutations. So that is the whole principle of targeted therapies. So dabrafenib, this is it is a, and vibrafenib, these are essentially against a protein called RAF, BRAF. These mutations are widely prevalent in skin cancer, that's melanomas. There are so many drugs like that. For example, there are inhibitors of SRC kinase, the c-Met gene, Herceptin. There are so many targeted inhibitors that target only this mutated or the altered signaling molecules. So this is an example of that. I mean, so again, another example, here is an EGFR mutation again. The mutation of EGF causes cell proliferation, invasion, angiogenesis, metastasis. There are very highly potent inhibitors of the EGFR signaling that prevents the downstream signaling. So what is the advantage of this targeted therapy? First of all, as I mentioned, it does not, or it is not supposed to kill the non-specific cancer cells. I mean, it is just does not kill a normal cell. So you can select the patient. So this will allow the personalized medicine. So this has been something that has been going on for the past many years, targeted cancer therapy. So you can take the cancer from the patients, they do a molecular analysis, and they find that some patients have genes in mutations, the blue gene, brown gene, yellow gene, green gene, etc. And you can tailor the therapy to the specific patient and for the specific mutations in the patient. So this has been fairly successful. There are so many of these drugs which are highly effective, and they have generally much less cytotoxicity than the regular chemotherapy drugs. Again, I am talking from the point of a pure cancer biologist, a pure cancer researcher, and not from a pure clinician point of view. I'm sure the clinicians, the cancer doctors will have slightly different opinion about their efficacy and how selective these guys are. So what has been the problem with the targeted therapy? So this is what I'll be talking about. I guess my Jisha or someone should tell me when my time is over so that I can stop and let you guys go.
It's okay, sir. It's okay, sir.
Yeah.
We have started 5 minutes. We're late today.
Okay, so just let me know. Oh, I have 2 minutes. I will stop at that time. So this is a problem with most of the targeted therapy. So this is a patient that was treated with vimorafenib with the BRAF mutation in a skin cancer patient. So the patient, this was what, before treatment, front and back, there are a lot of melanoma or the skin cancers. They were treated with this plexicon drug, that is vimorafenib, 15 weeks. They are completely cured of the cancer. So the patient has been sitting around with the drug. Within 23 weeks, the cancer came back with a vengeance. The patient had developed full resistance to this particular drug. So what is happening is that whenever you treat, or in most of the cases when people are using targeted therapies, especially the class of tyrosine kinase inhibitors, which is which are drugs that are targeted to tyrosine kinases, it works very well. But as I mentioned, the cancer cells are very smart. They evolve and they develop secondary mutations, or they develop bypass oncogenic pathways. They develop resistance to drugs. Invariably, any specific targeted therapy kinase inhibitors given to patients, they work beautifully for 9 months to 10 months, then the cancer comes back. So why does this happen? So that is a type of things which my lab has been working for the past 10, 15 years, even though I've been running a lab for almost 30 years now. So there are many issues. One of them is that the cancers are highly heterogeneous. I spoke about all the elimination of the mutated cancer cells, etc., etc. I mean, very nice pictures which we got from different places. But a problem is that cancers are not just homogeneous. Cancers are so many cell types within them. The tumor microenvironment, as I call, they have cancer cells, They have angiogenesis, they have endothelial cells and blood vessels, they have pericytes, they have immune cells, inflammatory cells, cancer-associated fibroblasts. Many of these cells promote the growth of these tumors. They also facilitate the invasion of cancer cells. So if you just kill one particular or a few of these cancer cells, there are these cells being promoted to grow by the surrounding cells. So the tumor microenvironment essentially contributes to the survival of the cancer cells. So it is very difficult to do this, to eliminate a cancer cell by itself without it coming back. So the microenvironment of a primary tumor, again, is very different from the microenvironment of a metastatic tumor that is already spread, because cancer from the lung, for example, goes to the liver, goes to the bone marrow. It's very, very different. So one other main reason is that this is something I would like to spend 1 minute on or 2 minutes on. So classically, what people believed that any particular cell in the body can acquire mutations, or any particular sensitive cell can acquire mutations, can divide uncontrollably. If they are not cleared by the immune system, they can divide uncontrollably, give rise to cancer, and this can acquire more mutations, give rise to cancer, etc. But what people found around in 1950s in the case of blood cancer, and around 20 years ago in year 2000 in the case of the solid tumors, is that only a subset of the cells within the cancer can initiate and propagate the growth of tumor. Only the subset of cells in the cancers have the ability to initiate tumors and also propagate the tumors. What people did was they took human tumors, dissociated them into single cell suspensions, put them in mice, and they found that around 1 to 2% of the cells can only initiate cancers in the mice from going from one mouse to the next to next. They called this as cancer stem cells. So now it is fairly established that these particular cells are the ones that initiate tumors and confer drug resistance and causing all the problems what I mentioned. So what are the basic functions of cancer stem cells? They are slow dividing cells that can initiate tumors. So they divide very slowly. So generally the chemotherapy agents cannot kill them, because chemotherapy agents target the fast dividing cells. They have the ability to generate heterogeneous tumors. They can not only give rise to cancer cells, they can also differentiate into other cell types within a cancer. Some of the cancer stem cells have the ability to metastasize to distal organs. Then they can remain dormant. So breast cancer, for example, can go to the lymph nodes, sit there around without any symptoms, and after 15 years, the dormant cells awake and they come back as highly drug resistant cancers. They are highly drug resistant. They have different metabolic and epigenetic signatures, and they are identified based on cell surface markers, etc., which I'm not going to talk to you too much about today because we are already running out of time. I just want to show you some of the things what you have done using this particular information we have on smoking-related cancer, especially lung cancer. As you know, smoking is highly correlated with the lung cancers. Tobacco carcinogens like NNK, NNN, they have been known to form adducts with DNA base pairs mutating vital genes like KRAS, P53RB. And recently, many people, including us, found that nicotine, they can function in a receptor-mediated fashion. And nicotine, which is the most addictive component of the tobacco smoke, itself is not carcinogenic, but it can promote cell proliferation, angiogenesis, and apoptosis. So nicotine is a tumor promoter. So this is an example of some of the studies we did around 10 years ago. We can implant tumors under the skin of the mice, give them nicotine by injection or by Nicoderm patches, which I think you are getting in India now, that as well as electronic cigarette, things like that. So this is just pure purified nicotine. The tumors are much larger and they are highly, highly metastatic. This is an example of EMT, what I mentioned, the normal cells are sitting around with very nice like kitchen tiles or their floor tiles with grout, everything sitting. You give them nicotine, you find that they change the shape, they undergo EMT, they become spindle shape, they can migrate. So nicotine can induce EMT, they can cause metastasis. Not only that, if you take the electronic cigarettes, which they have been telling that is a good way to quit smoking, and if you treat stem-like cells with the nicotine, you find that the stem-like cells can grow into these tumor-like spheres very well. So nicotine can essentially enhance the stemness of the stem-like cells and confer resistance. So the question is that what is driving these stem-like cells especially those are driven by nicotine and in lung cancer, and can you target them? To essentially make a very long story short in the next 3-4 minutes, there's a protein called SOX2, which was absolutely necessary for the growth of the stem cells. I mean, I'm showing 5 years of work in one slide. This protein is induced by nicotine in the stem cells as well as in other cells. And we found that SOX2 is induced by this protein called YAP1. YAP is a protein that is S-associated protein. So essentially, this YAP gene is altered in a huge number of human cancers. So if you can target YAP, that would be something very good. But YAP is a transcriptional coactivator. It does not bind to DNA. It does not have any enzymatic activity. So it is very difficult to target this particular protein. But what YAP1 does is that it binds to other proteins and promotes gene expression. Again, we are showing that people with high levels of YAP1 lung cancer patients, they die, whereas people with less amounts of YAP1, they survive longer. So we did multiple experiments to show that YAP is important for tumor growth. Here we are showing that nicotine and electronic cigarettes can induce YAP1 in the nucleus. So the question is, how do you target YAP1? We found that YAP1 associates with a protein called OCT4 through this particular domain of YAP binds to a second protein called OCT4. So we made peptides corresponding to this region. You deliver the peptide into cells and you prevent the growth of this stem-like cells here. So we started a drug discovery program to identify small molecule inhibitors of YAP1. We screened around 3,000 to 4,000 compounds, hardcore drug discovery protocols, which I can go through anybody if they can email me, or there's no need to go into the very high details. We are finding that our drugs can prevent the binding of YAP1 to OCT4, essentially suppressing SOX2, preventing stemness. The specific drugs will prevent so the red dots are gone, whereas inactive compound still have the interaction. So here we are finding that some of our earlier drugs, they can prevent the self-renewal of the stem-like cells or the formation of the tumor organoids. And we find that they can kill cells with very high potency. So again, as I mentioned, common mechanism of drug resistance, expression of multi-drug resistant proteins, amplification of drug target genes. There are multiple ways cells become drug resistant. So this is an example where we have taken the stem-like cells, grew them in a classic chemotherapy agent, cisplatin. The stem cells are not sensitive to cisplatin, they continue to grow. Now you take these cells which are resistant to this chemotherapy drug, and you grow them in the presence of our drug, the YAP inhibitor, and we find that they are completely eliminate the growth of this drug, of this compound. So by targeting stem cells, you can overcome the drug-resistant growth of stem-like cells and perhaps the tumors. Those are some of the type of studies we are doing. So here we are finding indeed the drug can eliminate drug-resistant cells. So again, as I mentioned, one of the major problems of chemotherapy agents is that they kill the normal cells in the body, and that is causing a lot of the side effects. So we did an experiment to see whether these YAP inhibitors are targeting the normal cells. So we took cancer cells, stained them with a red dye, normal cancer-associated fibroblasts, stained them with a green dye. We grew them for 48 hours. You can see the cancer cells take over the co-culture. Now you take this co-culture and treat them with a drug. You can see that after 48 hours, the red cells do not grow, the cancer cells do not grow very well, whereas the green cells or the normal cells are growing very effectively, suggesting that these drugs can essentially eliminate the cancer cells with very little effect on the normal cells, showing that it can preferably kill the cancer cells by eliminating YAP1. So this is a lung cancer organoid experiment. You can take the actual tumors from patient and grow them as organoids, grow them as small bits in specific media. So this is probably the best model to study cancer outside a mouse. And if you take this lung cancer organoids, probably I'll go to the next slide. You can see that the green cells are the live cells. Our drugs can completely eliminate the growth of the lung cancer organ. So these are the small lung cancer samples from patients which we are growing in culture. So it has all the microenvironment components, etc. And you are completely eliminating this by inhibiting YAP function. So these are some of the inactive compounds what we had used. So this is a live dead assay where what we call, where you can, in the same experiment, you can estimate the live cells versus the dead cells. So essentially we are finding that you can target the cancer stem-like cells, which are very important and for the maintenance of maintenance and recurrence of growth. So we have some very good drugs which we have developed in the lab, but the issue is that, as people know, a drug from the lab to come to the patients, it can take anywhere from 5 to 6 years. So just doing this in vitro work, what I showed in the tissue culture dish, that may not be fully effective. The drugs could be very unstable in the body. They may not be very efficacious. It can be excreted very fast. Then it may have many, it should have real drug-like properties, and that is pretty challenging. And that's what we are trying to do right now, to make these inhibitors as proper drugs that can work selectively. So because of that, we have done other experiments trying to find other inhibitors of YAP, and we have certain other compounds where we find which can again inhibit YAP very effectively by inhibiting arginine methylation of YAP1, which is a brand new story which we are hopefully going to publish very soon. This is another class of inhibitors which our lab is developing to combat YAP1 and to eliminate stem-like cells. So this is again looking at a tumor section, multispectral imaging. We can stain for up to 7 or 8 proteins at the same time. We find that wherever YAP is there is higher levels of SOX2 in the stem-like cells. Arginine methylation proteins are high with YAP, etc. So targeting this particular protein is something our lab is trying to do to eliminate the cancer stem-like cells. And YAP also acts as an inhibitor of T cell function. So if you can inhibit YAP and reduce YAP levels, it can actually enhance immune response as well. So that is a totally different angle we are looking at. And I think that's a story for another day. So this we are showing lung cancer organoids. If you inhibit YAP using our second type of inhibitor, the Korman inhibitor, you can reduce the SOX2 expression. You can reduce YAP levels. You can inhibit tumor growth, etc. So these are the people who did a lot of the work in the lab. Mohan, Namrata, Vishwarupa, Jaya, and Fatima is doing a lot of work now. These are some of the previous members in the lab. These are some of our collaborators, and we have been very lucky to obtain funding from National Cancer Institute, DOD. A company had licensed our drug in the past, and they helped our project as well. So I think I will stop here and I will take any questions you may have. Sorry for going over time, but there are so much stuff to talk about. I will stop there and take questions. And if you don't mind, I can stop sharing the screen.
Thank you, sir. Excellent talk, sir. We are lucky to have you today.
Thank you. It was very nice of you to invite me, Jisha. It's very nice of you to invite me.
And we can move into the Q&A session. Sir, could you see the Q&A box on the lower panel? Shall I read the questions one by one?
Yes. So you want me to read it and answer it?
Your choice, sir.
Yes. Yeah, so Sandra Marie has asked if malignant melanoma is a severe case of cancer, and why did the symptoms of this appear on nails? So malignant melanoma, yes, it is a serious type of cancer. There are, as I mentioned, some BRAF inhibitors. Angane chala therapyകളുണ്ട്, വളരെ effective ആയിട്ടുള്ള therapyകളുണ്ട്. Immunotherapy also works for melanoma. So if the patient is have PD-L1 level, CTLA4 level, etc., he may respond very well. So there are different types of melanomas. So there is acral melanoma, which happens on the whites of kayyada vellayilum, kaalinte vellayilum, nagathilum varuna acral melanoma, the type of cancer. There's uveal melanoma, which is a skin cancer that happens in the eye, inside the eye. There are different types of melanomas. And that can be a pretty severe case of cancer, but if detected early, they can all be cured. What is HPV? Why does it occur? Which variant is harmless? What are the measures to avoid HPV? I know homemade remedies. I don't think there are any homemade remedies for any type of cancer. I mean, you can improve your lifestyle, eat organic food, etc., etc. Pesticides, herbicides, food. Otherwise, please don't believe that any of the normal homemade therapy will cure or even improve cancer. Nope, it does not. So HPV, the human papillomavirus, it is a virus that causes cervical cancer as well as oral cancers. It has two specific oncogenes, and the HPV 16 and HPV 17. അത് രണ്ടുമാണ് പ്രധാനമായിട്ട് ആക്റ്റീവ് ആയിട്ടുള്ള വൈറസുകൾ. So it has some specific E6 protein, E7 protein, and certain proteins on the which can essentially inhibit many of the tumor suppressors. HPV E7 protein inhibits the RB tumor suppressor gene, as well as E6 protein will inhibit the P53. This protein will eliminate him, so it will essentially inactivate the tumor suppressor genes by viral means rather than by a genetic means. So HPV may not be too big of an issue going forward because there are antibodies, there are vaccines against HPV now, Gardasil nallam paranja pala vaccines undu. So I think the HPV mediated cancers may go down in the coming years because there are effective, people can be effectively vaccinated against that. There are several serotypes of this HPV that is harmless, which they don't cause, and they may cause warts and all that. There are nothing. They are not cancerous. So there are a lot of advance in the HPV mediated therapy. So Karthik has asked, cancer is not formed in our heart. If it is true, what was the reason for that? I mean, those are very, very rare. So one of the major probable reason for such things not happening is that cancer, the heart tissues are highly differentiated tissues. And not only that, the cardiac muscles have a totally different metabolic mechanisms or metabolic events compared to a normal cell in the body. So for whatever I know, there are the biology of the cardiac muscle cells, how stringently how strongly differentiated those cells are, and their metabolic signatures, the metabolic pathways they follow, make it very different for cancer to grow, or for those cells to become a cancer cell. It is very, very difficult. So that is why the incidence of those kind of cancers are extremely, extremely low. So Ramakrishnan has asked, thank you for the comment. Up to which stage can cancer be cured completely? Is it possible to cure patient that one has reached the metastatic stage? So metastatic cancers are always touch and go. I mean, if it has not spread, if it is still localized, the chances of survival are extremely, extremely high. But for pretty much for any type of cancer. But the problem is that diseases like pancreatic cancer, people have absolutely no symptoms at all. In the end, people have severe stomach pain, things like that. They go to a doctor. By that time, they find that the cancer is already spread to liver all over the body, and it is way too late. So most of the cancers, if they are highly metastatic, it is difficult to cure. But the chemotherapy and especially the immunotherapy development that has happened in the recent years, those are all very promising approach to have to combat metastasis. And again, as I mentioned, targeting the stem-like cell, the cancer stem cell, that could be another approach. Metastasis vannal pinne namukku systemic therapy pattathullu. We cannot do radiotherapy or surgery or anything like that. So that is a major problem. And as I mentioned, all these cancers can develop resistance, but all these therapies can extend your lifespan for a cancer patient. Again, depending on the cancer type, what stage it is, it's very difficult to say. If you have a stage 3 cancer, you are going to be cured, whether it is melanoma or breast cancer or whatever. It depends on the patient to patient, the type of mutations we have. But if it has spread, it is difficult. It is a challenge. You have to manage so many different things. In India, the percentage of cancer patients are enhancing year by year. Let me the reasons for this occurrence. I mean, it is very interesting. There are a lot of lifestyle matters, a lot of lifestyle changes, a lot of environmental factors. I told that cancer is a genetic disease and an epigenetic disease, etc. But if you do a search for cancer causes, just do a Google search. One of the few things that come up is your diet, smoking, environment, etc., etc. So all these things can cause cancer, is all contribute to the genesis of cancer. For example, in India, for the vehicles, the number of vehicles, automobiles have gone high so much. There's so much pollution. A lot of the food, you have herbicides, pesticides, a lot of adulteration. All these things can cause to the enhanced level of cancers. The number of trees are less. I mean, it is very different from what it was 30 or 40 years ago. All those things contribute to it. I should also mention that, I mean, these environments play a major role or contribute significantly to the genesis of cancer. One interesting finding is that if you go and live in a different environment, for example, so I hope it never happens, but if I develop cancer, I will be developing a cancer that is more prevalent in the US, even though genetically I am 110% Indian. Similarly, if somebody from US goes and lives in Mexico or comes and lives in India, they are more prone to get cancers that are prevalent in that area, that region. That is because of the diet, the environment, etc., etc. So the environment, the diet, everything contributes the type of cancer you are getting, type of mutants you are getting, what is the environment, all those things play a major role in that. So it is very difficult to enumerate. This is a particular reason. This could be reason. I mean, olden days, the DDT and all that cause a lot of problems, developmental problems. Smoking, it has been very well demonstrated to be oncogenic. So lifestyle changes, eating good food, being more careful, and try to stay healthy. I mean, don't end up looking like me, the Kodavira and all that. Try to be, I mean, healthy and active. That will be a very good deterrent against cancer. Why cancer won't affect cancer areas? I think in the could be metabolic. The cells are highly differentiated, etc. That could be one of the major reasons. What is the future of oncolytic virus treatment? Is it better than chemotherapy? In which case, treatment were gone. So there were a lot of trials in the past on using oncolytic viruses, especially in brain cancers, glioblastomas, neuroblastomas, etc. So in the clinical trials, they were supposed to be very successful. But I don't know there is how much excitement is there for the oncolytic virus therapy nowadays, because people develop different complications and the viruses have to be developed into the brain, etc. It is still a possibility, but people don't know whether it has a distinct advantage over the immunotherapy or other modes of therapy that are currently being used. But it may come back up again. We never know. Technologies are developing, things are developing. The COVID vaccines is a typical example how fast they're developed, how they use mRNA vaccines that have never been used before, and it is how effective it is now. So those kind of vaccines probably perhaps can be developed against oncology, against HPV, things like that, HPV caused cancers, or even regular cancers with the mutants. There are a lot of possibilities going forward, but as of now, oncolytic viruses are not the most exciting or the most attractive candidates for cancer therapy for various reasons. So most of the people, this is from Kartika, detect the cancer at very late stage. How can we detect them if it is in very early stage? What are the earliest symptoms? See, that is a problem. People don't show symptoms till the cancer is fairly advanced. That's a major problem. So how can you detect cancer early? I mean, if you have fever that doesn't go away for 2 weeks or something like that, so light fever, I mean, it's COVID times, that could be other things too. But if you have fever and cold and cough that doesn't go for some time, or regular doctor visits. Here in the US, you are required to see your primary care physician at least once a year. And if you have any problem, you're supposed to see every 6 months. You are required to do regular checkups, regular monitoring, especially for women, mammography, pap smears once you're a certain age. Again, cancer is a disease of the older age. So essentially once you're in the 40s and 50s, do colonoscopy, do your screening, the skin cancer screening, etc. One should do. That is the only way to do it. There are no symptoms you can say that can happen. I mean, as I mentioned, pancreatic cancer, for example, is a very aggressive cancer. There are hardly any symptoms. It just hits you like a car coming and hitting you. So it's very difficult to predict. What is the link between hematological malignancies and increased risk of COVID especially? So this is Manisha's question. So they have shown in the earlier studies that people with leukemia and lymphoma, they are generally more prone to COVID, and those patients are essentially having more severe symptoms of COVID. I mean, essentially they are having the more severe cases. That is because, as you can imagine, the heme malignancies like leukemia and lymphoma, they are being they are affecting the B cells, the T cells, and other cells of the immune system. So if they are altered, their immune system is compromised already, and that gives it a much more conducive environment for the virus to grow. So it is, and they all get systemic treatment. They all get chemotherapy treatment or similar treatments for these blood cancer. So obviously those people are a little bit more prone to COVID than, or they have more severe cases of COVID than other one. പക്ഷെങ്കിൽ അതിൽ ഈ റീസെന്റ് സ്റ്റഡീസിൽ പറയുന്നത് അതിൽ അങ്ങ് സ്റ്റാറ്റിസ്റ്റിക്കലി വളരെ സിഗ്നിഫിക്കൻ്റ് ആയിട്ടുള്ള വ്യത്യാസമില്ല എന്നാണ് പറയുന്നത്. സോ don't stop the cancer therapy. Those patients shouldn't stop because of afraid of COVID or severity of COVID disease. Just continue the disease. That's what they are saying. Well, Karthika has asked another question, which is, which is cellular aging. Does it have any relationship with cancer or cancer treatment procedures? Cellular aging, yes, definitely it has a correlation with cancer. Essentially, as I mentioned, cancer is an old people's disease. So people have more senescent cells they have. It contributes to cancer. It contributes to drug resistance. But unfortunately, there are no medicines that can contribute senescence, or people like me will find it very useful. So there is no way to, so far, to keep the cells very young. So as the cells divide and the progress and the person gets older, the chances of accumulating the mutations are much higher. So there is definitely higher risk of cancer in older patients or patients who have more older cells in their body. But it does not affect treatment procedures. I mean, you treat the cancer rather than saying, okay, this person is, the cancer treatment is generally, you are not just treating the cancer, you are treating the whole patient, looking at the rest of their health, especially if you are old, how much can you handle, etc. So the age comes into play, but cellular aging, that itself is it's a huge field. If you do a search on PubMed, cellular senescence and cancer, you will get at least a few thousand papers. So of course, there is definitely a correlation there. Anonymous attendee, ഇന്ന് നമ്മൾ ഓരോരുത്തരും മൊബൈൽ അമിതമായി ഉപയോഗിക്കണം എന്നുള്ള യാതൊരു സംശയമില്ല. മൊബൈൽ റേഡിയേഷൻ ക്യാൻസർ വരാൻ കാര്യമാണോ? അത് ഡോക്ടർ സൂര്യ. So far അതിന് യാതൊരു തെളിവുമില്ല. There is ആരോ ഫോളോ ഓൺ ചെയ്തിട്ടുണ്ട്, അതാണ്. And I think people got current now. Good. ഇതിൽ there is no evidence to suggest that there's no strong correlation. മൊബൈൽ ഉപയോഗിക്കൊണ്ട് ക്യാൻസറുമായിട്ട്, അതിനെക്കാട്ടി ബാക്കി അസുഖങ്ങൾ വരാനാണ് ചെവിക്കുള്ള പ്രോബ്ലവും കണ്ണിനുള്ള പ്രോബ്ലവും കൈക്കുള്ള പ്രോബ്ലവും എല്ലാമാണ് കൂടുതലും ചാൻസ്. ക്യാൻസറിന് അതിനെപ്പറ്റി, അതും ഈ 5G നെറ്റ്വർക്കിനെ പറ്റി ഒന്നും there is no evidence so far. ഇനിയും വരില്ല എന്നല്ല, so far, as of today, as of now, there is no strong evidence, but things can change. That's how science works. So ഇതുവരെ യാതൊരു ഇതുമില്ല. How long does it take a normal cell to acquire the appropriate type and number of mutations to become a malignant cell? Aniket Kashyap ചോദിച്ചേക്കാണ്. So ഈ ട്യൂമർ സപ്രസർ ജീൻസിലാണെങ്കിൽ ഒരു സെല്ല് ക്യാൻസറസ് ആകണമെങ്കിൽ you need to have 2 mutations in both the alleles. Essentially ഇപ്പോൾ RB ആണെങ്കിൽ റെറ്റിനോപ്ലാസ്റ്റോമ അല്ലെ P53 ജീൻ ആണെങ്കിൽ അതിൻ്റെ ഓരോ ജീൻ്റെ 2 അല്ലീലല്ലേ ഓരോ. So both the alleles have to be mutated. So generally what happens is that people acquire one mutation from their parent genetically and one mutation happens somatically because of their exposure. That is called a 2-hit hypothesis. Alfred Knudsen, that's one of the famous hypotheses. For the oncogenes, pretty much the same. Oncogene 2-hit in the avishya, even one mutation and leads to the overexpression can cause cancer. Again, it depends on just because a cell acquired a mutation doesn't mean that it is becoming cancerous. It has a potential to become cancerous. It has to be in the right environment. It should have the right supporting cells. It should be able to invade the immune system, etc. എന്നാലേ അത് ക്യാൻസർ ആകത്തുള്ളൂ. Just because RB gene is mutated, you have some chance. It has a much increased chance. പക്ഷെങ്കിൽ അത് തന്നെ കൊണ്ട് ആകണമെന്നില്ല. So generally it is how long does it take എന്നുള്ളത് പറയാൻ വ്യത്യാസമുണ്ട്. Depends on how fast the cells are dividing in the body. ഇപ്പോൾ നമ്മുടെ സ്കിൻ സെൽസ് ആണെങ്കിൽ ഔട്ടർ സെൽസ് എല്ലാം they don't divide at all. Some of the cells that divide, they have mutation, they can develop, but it can take years actually for a cancer to develop. So when people are diagnosed with cancers, what they should remember is that it has been growing for a long time. It takes time to develop into cancer. Explain about T cell transplantation. So T cell transplantation people have been doing, especially CAR T cells. T cells, it's a modify either you try to express another receptor in it or another and put it back into patients. So there is a huge amount of literature on that. അത് പക്ഷെങ്കിൽ കൂടുതലായിട്ടും വർക്ക് ചെയ്യുന്നത് ഇൻ ഹീമറ്റോളജിക്കൽ മാലിഗ്നൻസീസ്, അതായത് ബ്ലഡ് ക്യാൻസേഴ്സിന് അത് കൂടുതലായിട്ട് വർക്ക് ചെയ്യുന്നത്. ആൻഡ് മോഫിറ്റ് ഈസ് വൺ ഓഫ് ദ leading centers in that. I cannot go too much details of that. There is chimeric antigen receptor, CAR T therapy. Antigen receptor, chimeric T cell receptor, chimera undaki. They isolate the patient's T cells. They express the chimeric antigen receptor in the T cells, grow the T cells in culture, and put it back in patients. And these attack the cancer cells and eliminate them. That is very expensive, but it is very effective in the case of many hematological malignancies. I mean, these are all very interesting questions. I mean, I wish I had more time, but I have a lot more questions here. Is there any correlation between social profile of the people and the incidence of cancer? This is Vincent Vijan Chochaiknana. I mean, they are very, very good questions, actually. I mean, and he's from the Department of Economics, so it is very relevant, actually. Heterogeneous religious communities living in our society, and they are following a little bit different lifestyle and food habits. Is there any cause and effect related between lifestyle and incidence of cancer? Yeah, there is a very good correlation between, I don't know about India per se, but the socioeconomic status and the occurrence of cancer. Especially in the US, I can say there is a big disparity in the occurrence and survival of cancer patients depending on your socioeconomic status and your race. The African American, അതായത് നമ്മളുടെ African Americans, കറുത്ത ആൾക്കാർ, നമ്മുടെ Hispanic population, those are the Spanish speaking people, especially from Mexico and others who live in this country, they have a much higher incidence of cancer, much lower rate of survival, etc. Part of it could be racial, depending on the genes what they have. Part of it because of their lower access to healthcare, their lower economic status, etc. But there is a very strong correlation between the race, the socioeconomic status, and the incidence of cancer. Exactly, the diet, everything matters. So it is a very major area of research, actually, and it's a very important area of research. I mean, even especially from the economists and the population biologists, it is absolutely important. Junio, what is erlotinib? Erlotinib is an EGFR inhibitor. Lung cancer, EGFR gene is mutated in many of the lung cancer patients. Erlotinib used to work very, very well. As I mentioned, after 9 months, every single patient developed resistance to that. That is because the patients who are treated with erlotinib usually acquire a second mutation called a gatekeeper mutation. So cancer cells, as I said, cancer cells are very smart. They grow, they divide, and you are treating with one drug, erlotinib. So some of the cancer cells mutate the EGFR in a different way, and they call it as a gatekeeper mutation, and they prevent erlotinib from binding to the mutated EGFR. That's how patients mainly develop. There are multiple mechanisms that develop resistance to erlotinib. അതുകൊണ്ട് ഇപ്പോൾ വേറൊരു ഡ്രഗാണ് EGFR mutant patients ഉപയോഗിക്കുന്നത്. It is called osimertinib. Osimertinib എന്ന് പറയുന്ന ഒരു ഡ്രഗാണ്. That is the one that is nowadays used for treating this EGFR mutant lung cancer. So it is the EGFR inhibitor. Why do some Aniket ചോദിക്കേണ്ട, why are some of the disseminated tumor cells proliferate to become clinically evident metastasis, some do not? That just depends on the tumor, and that just depends on the person pretty much, how aggressive the tumors are, what are the mutations being present. I mean, is a person obese? It could be a lot of other conditions that we don't know yet why it is happening. It could even the dormancy, especially breast cancer patients and to certain extent lung cancer patients, you get all these small metastatic foci sitting around in the bone marrow and the lymph nodes without any symptom. After 15-20 years after you are recovered from it, these guys come back up. It depends on the changes in the body physiology, what is the hormone level, the cytokine levels, etc. So nobody really knows about that. I am a PhD holder, in history. Can I study MBBS? Is there any option? Actually, if you go out of India, you can do that, especially in this country and many other countries. If you can study biology on your own, you will have to pass the entrance exam equivalent. You can do MBBS.
Maybe inspired after your talk, sir.
I doubt it. I really doubt it. But anyway, these are all very important and very, very interesting questions, actually. And especially, I'm pleased to see questions coming from people outside the science background as well. Karthik, I say you very little, but this topic is more learned. Is cancer more common in female sex? How sex take part in cancer? It depends on the tumor type, actually. It's more common in females, especially nowadays. There's prevalence of breast cancer is coming up because lung cancer is being handled much more better with immunotherapy and other ways. So the female hormones and female and the uterus, ovarian cancer, breast cancer, etc. is higher in one, but other than that, they are very similar, but females have a slightly higher rate nowadays, especially coming to breast cancer. But hopefully that will change. If you are giving the same talk 5 years from now, I mean, I'm sure I'll be still working 5 years from now. I'm sure it may be the whole dynamics and the numbers of the patients and the type of cancer will be very different in the coming 5 years. So just because you are female, you don't have to worry. I mean, it is comparable to men only. Is there life expectancy of different tumor cells? No, essentially tumor cells can live forever. I mean, the HeLa cells, there is a book called the Henrietta Lacks and a book about the HeLa cells, the immortal life of Henrietta Lacks on a book. So the cancer cells can be grown forever in a tissue culture dish. So there is no, that is, so there are if you Google the hallmarks of cancer, one of the hallmark of cancer is the cells have uncontrolled cell proliferation. They can live forever. One of the hallmarks. So another normal cell ആണ് ഇപ്പൊ നമ്മൾ കൾച്ചർ ചെയ്താൽ ഇപ്പൊ സ്കിന്നിന്റെയോ അല്ലെ വായിന്നോ എല്ലാം എടുത്ത് കൾച്ചർ ചെയ്താൽ it will after 8 to 10 generations they die. They don't grow beyond that. The cancer cells can keep on going and keep on going. So there is no life expectancy. Anonymous certainty, in fact, COVID-19 kind of virus enhance the kind to proliferate rapidly. So this person is asking whether COVID-19 virus, can it enhance a cancerous cell to proliferate rapidly? There is no evidence for that. There is absolutely no evidence that the COVID virus, the coronavirus can enhance cell proliferation. It is not an oncovirus. Oncogenic virus ധാരാളം ഉണ്ട്. HPV നേരത്തെ ആര് ചോദിച്ചാൽ adenovirus അങ്ങനെ പക്ഷേ ഇത് അതുപോലത്തെ വൈറസ് അല്ല. It does not cause cell proliferation. Sir, how can we do higher studies in this field? There are so many fantastic institutes in India. Our Rajiv Gandhi Institute in Trivandrum, നമ്മളുടെ Sri Thirunal Center. There are so many Kerala University. There are so many fantastic places where you can do PhD or masters related to cancer therapy and cancer biology. In Kerala or in all over India, the status of science has improved so very much. There are so many opportunities. If ever you are interested, I mean, I strongly encourage you. It will be something very relevant to the people there. Why the doctors recommend Lily Devi treatment of radiation oncology as a patient? Why is it preferable to others? So essentially, the tumor is localized, is spread ചെയ്തിട്ടില്ലെങ്കിൽ ഞാൻ പറഞ്ഞ പോലെ the best options are surgery or radiation. If radiation is applicable, if the person responds to that, that is probably the best way. Invasion ഒന്നുമില്ല, അതായത് നമ്മൾ ശരീരം ഒന്നും മുറിക്കുകയൊന്നും വേണ്ട, but there will be side effects. And radiation oncologists are the doctors which specialize in giving radiation. If you look at principles of radiotherapy in the Google, you will be amazed at the amount of mathematics, the physics, the science involved in giving radiation therapy. It is so important to get the right dose to the right area of the patient without affecting the rest of the body. So a radiation oncology is absolutely necessary. And if a patient can be cured by radiation, if somebody has recommended someone you know to go and see a radiation oncologist, he should definitely do that because it is highly promising. Osteosarcoma on the knee after surgical removal, does it reappear again in later years? Manju Subramania. Yaduru guarantee there is no reason for it to reappear if it has been completely removed and if the person has been cured for a long time, it is completely gone. But you never know. In the cancer, one can never say what is happening in one person is very different from the next person for the same type of cancer. based on the genetic makeup, based on the lifestyle, everything. So we don't know, and the age. But osteosarcoma എല്ലാം വലിയ കുഴപ്പമില്ലാത്തതാണ്. അത് usually if it has surgically removed, I mean, it can reappear in other parts, etc. But the same part, യാതൊരു കാര്യമില്ല. Vishwarup Singh Yodikanda, is there any way or chances to produce vaccines to stop certain cancers? Yeah, there's a huge effort to develop vaccines against cancer, but it is a slow field. There are some vaccines, but they're not very effective. But again, the problem is that cancers are like growing an extra finger, or it is your part of the body that is growing inside you. So it is very difficult to have an antibody that targets just that part of the body and not other parts of the body. So it is difficult. How does cancer kill the patient? I mean, Aniket Kashyap, it does so many things to you. It shuts down the system. It can prevent it can totally alter the metabolism. It again feeds on the cells of the body and alters the whole metabolism and everything. I mean, you can read about that. I mean, there are so many things that happens. What are the probable causes for thyroid cancer? Dr. Shija MS. Probable causes, I mean, that is very difficult to say. It is probably genetic, especially in India. Thyroid cancer is fairly common. I think even here it is there, very fairly common. The probable causes, it is all genetic and lifestyle. It's very difficult to say why that is. But one thing I can say is that thyroid cancer is one of the most curable cancers out there. So if somebody has a choice of getting a specific type of cancer, your neighbors, family, whoever, thyroid cancer will be the best cancer to have because it can be completely cured. 95% of cures. I mean, so it is a highly curable cancer, but I don't think it can be prevented. Like most of them. Whether a couple with Tumor is longing for death, I had no idea what Ginu is asking. Chemotherapy is necessary in all cases after mastectomy. No, that depends on the somebody has is chemotherapy and radiation necessary in all cases after mastectomy? So after mastectomy, the reason why they suggest chemotherapy and radiation is that sometimes many of the cells are left at the site of the surgery. The radiation can remove that. Why they suggest chemotherapy is that surgery നടത്തുമ്പോൾ, especially in breast and all that, some of the cancer cells can get dispersed in the body and they may be already circulating. And also, even though there are no symptoms of spreading, even the localized cancers, there may be cells circulating in the body, in the bloodstream. So the chemotherapy will eliminate all the circulating cells, clean up the body, and eliminate all the cancer cells. That is why it is happening. Sojamon Matthew, why body weight in a cancer patient decreases drastically? It is called cancer cachexia. The cancer cells, so it feeds on itself. Essentially, the cancer cells cannibalize other cells, the energy metabolism, it feeds on the other cells and eliminates a lot of the cells in the body. That's how you lose your body mass. That's one of the major ways by which cancer eventually causes death. You cannot eat because of the effects of the chemotherapy on the alimentary canal, etc. And അതെല്ലാം കൊണ്ടാണ് എൻ്റെ പേഷ്യൻസിന് ഒരുപാട് പ്രശ്നങ്ങൾ വരുന്നത്. Any scope of eliminating the oncogenes using CRISPR technology, Dr. K.C. Rajan? It may be eventually in the future, not immediately. എന്ന് പറഞ്ഞാൽ അത് from the body, oncogenes play normal functions in the normal cell. They all play functions. Only when it gets mutated, it has problems. So eventually in the future, it may be, but as of now, it is difficult to. But people are attempting CRISPR technology for some other genetic diseases where it is already inherited, where you can eliminate the genes. So that is fine. Aniket Kashyap, do metastatic cancers undergo a period of dormancy? Many of them do, and many of the metastatic cancer cells stay dormant for a long time before they grow up. So that is something highly, highly possible. So they can do that. What is tongue trauma? How can be issued? Ginu, tongue trauma, I have no idea. That is not directly related to cancer. Rajkumar, can a cancer patient disposable for COVID vaccination? Yes, exactly. They can take COVID vaccines, especially if they don't have a too much problem. I mean, with other health-related problem, they can take the COVID vaccine. It will be very good if they don't get COVID. Anything that you can prevent them from getting COVID, it will be. it will be very useful, and it should be strongly be encouraged. They should get the COVID vaccine if they can physically handle it. I think that is the end of the questions. I think I spoke for way too long. So if there are no more questions, I think I can stop there. I guess everybody has gone to sleep by now. It is pretty late there, right? 8:30. So with your permission, maybe I can log off. Jisha?
Hello, sir.
Parayu.
Sir, my internet was a bit slow.
Oh, I was wondering what was that. Okay.
Shall we wind up, sir?
Yes, of course.
It was like a PSC question. It's
Indo?
series of questions. Divya?
Teacher, okay.
Okay. Now I invite Dr. Divya PS, Assistant Professor of Zoology, SN College, Kollam, for the formal out of thanks.
Thank you, Jisha teacher. Good evening to all. Here we come to the end of the 25th session of this International Multidisciplinary Webinar Series, Agora 2021 by Sri Narayana College, Kollam, in association